G1 is the first period of the cell cycle. During this phase, the cell grows in preparation for DNA replication. The cellular environment and the cell size are monitored during this phase to ensure that the conditions are suitable enough to support the DNA replication. The cell cannot continue on in the cell cycle until it is ready, and in the event that the cell is not ready, it will enter a phase called G0. When the cell has an environment that is appropriate enough to allow it to move forward in the cycle, it will advance to the next stage of interphase, which is called synthesis.
G0 phase is where the cell is neither dividing nor preparing to divide. Cells enter the G0 phase from a cell cycle checkpoint in G1. During G0, the cell cycle machinery is dismantled and cyclins and cyclin-dependent kinases disappear. Cells then remain in G0 until there is a reason for them to divide. Although many cells in G0 may die along with the organism, not all cells that enter G0 are destined to die. This is simply a consequence of the cells lacking any stimulation to re-enter in the cell cycle.
S or synthesis phase occurs after the G1 phase. This is the portion of interphase during which DNA and centrosomes are replicated and the total number of chromatin are doubled. Chromosomes are copied and duplicated only in this phase. This phase involves the use of S-phase cyclin-dependent kinases and S-phase cyclins. The phase begins after the cell has gathered the nutrients, energy, and size to pass the checkpoint between G1 and S phase. The nucleus prepares the cell to duplicate its DNA and centrosomes. As the replication process continues, cyclin E is destroyed, and the level of mitotic cyclins begins to rise as the cell enters G2 phase.
G2 is an intermediate phase, a time for the cell to ensure that it is ready to proceed into the cell cycle. G2 can be thought as a safety gap during which a cell can check to make sure that the entirety of its DNA and other intracellular components have been properly duplicated. If not, G2 can actually arrest the cell and prevent its entrance into mitosis by inhibiting a protein called cell division cycle protein during the G2 checkpoint. G2 is also the cell's final chance to grow before it's split into two daughter cells during mitosis.
Prophase, the first and longest part of mitosis, begins when the chromosomes have already been duplicated. The chromosomes begin to coil and both become shorter and thicker. The nucleus also begins to disappear. In the cytoplasm, the mitotic spindle forms between the two pairs of centrioles as they migrate to opposite poles of the cell. The nuclear envelope disappears at the end of this phase, allowing it to enter the nucleus.
During metaphase, the centrioles start at opposite poles in the cell. The pairs of homologous chromosomes become arranged on a plane called the metaphase plate. Spindle fibers from one pole of the cell attach to one chromosome of each pair. Spindle fibers from the opposite pole attach to the homologous chromosome.
Entrance into anaphase is triggered by the inactivation of M-phase promoting factor that follows mitotic cyclin degradation. During anaphase, the kinetochore microtubules retract and separate sister chromatids to opposite poles. This process is known as anaphase A. The mitotic poles marked by these centrosomes themselves separate by the elongation of a specific type of non-kinetochore microtubule called a polar microtubule. This process is known as anaphase B. In the end, there are now two sets of chromosomes on either side of the cell.
In telophase, the sister chromatids reach opposite poles. The small nuclear vesicles in the cell begin to reform around the group of chromosomes at each end. As the nuclear envelope reforms by associating with the chromosomes, two nuclei are created in one cell. The chromosomes begin to decondense and become more diffused as well. Cytokinesis is the process in which the cell actually divides into two. With the two nuclei already at opposite poles of the cell cytoplasm separates and the cell pinches in the middle, ultimately leading to a cleavage. In most cells, the mitotic spindle determines the site where the cell will begin to split.
In the event that a genetic mutation causes a regulatory protein to malfunction, the G2 phase loses its checkpoint. Without the G2 checkpoint, growing cells will rapidly enter mitosis, even if they are not suited. This rapid production of mitosis-bound cells can result in a clump of cells known as a tumor. In most cases, these tumors or cancers can lead to health risks.